Hello and welcome to Nikolai's genetics lessons, and today I have prepared two very easy questions for you, and as usual I recommend you to pause video here, read the questions, choose your correct answers, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. So here is the first question: which of these is an example of the stem cell? And here is the four answers to choose from. Answer A, a red blood cell. Actually, red blood cell cannot reproduce by itself. Red blood cell even doesn't have nucleus, so it cannot reproduce. So red blood cells even cannot have adult stem cells, and adult stem cells can be found throughout our bodies. For example, if you cut your skin, it would be healed, of course, and we also shed some of our skin cells every day, so we do have such called adult stem cells throughout our body to replenish those cells that die. You can exclude this variant, and answer B, neutron. Neutrons can have adult stem cells, so this could be possible answer. And for example, answer D, skin cells that reproduce new epithelial cells, as I said, we also can consider as a correct answer, because these cells has stem cells that reproduce new cells, but they have some limitations that they can reproduce only in those cell lines in which they found. So let's consider answer C, a cell from a blastocyst. So what is a blastocyst? Imagine that we have a sperm and we have an egg. Egg is much bigger than sperm, and when they would join, we would have blastocyst. Blastocyst start to divide, so we have more and more cells with each division. So we call this at the early stage early blastocyst, that would later develop into the late stage blastocyst. So it would look like this. Here we would have a layer of cells. So as you see, some of the cells start to differentiate here, and also we can find here a clumps of cells that we call embryoblast, and cavity here we call blastocyst cavity, and this outer layer of cells we call trophoblast, and trophoblast cells would become placenta later. So as you see here, we already have at this stage some differentiation, and of course these cells here can differentiate into any other type of the cells, and of course at this stage also, but later most of them would become differentiated cells and wouldn't be able to differentiate in any type of the cells, only could become the cells from which cell lines they arise. So as you see, the universal stem cells could be blastocyst cells that can become any type of the cells. Once again, so-called adult stem cells can be found in any tissues, even of the adult, and the younger the person, the more such cells we can find in its tissues, and the older, for example, man becomes, the fewer such cells we can find. That's explanation why old people need more time to heal their wounds, for example. So the correct answer here would be answer C. And next question: knockout mice can serve as model organisms for study of human disease. is it true or false? And the truth is that we humans share more than 90% of the genes with mice. Those we are so distantly related organisms, but still we have many genes that have very similar sequence, and that means if a gene has very similar sequence, it would produce very similar proteins, and similar proteins would be involved in the similar biological pathway, so would perform similar functions. That means when we knock out some of the genes, and how we do it, imagine that here is a sequence of the DNA that code for some protein. For example, it could be A, T, A, C, G, G, A. And as you know, 3 nucleotides code for the amino acids. So as you see, here we have 2 codons, and if we, for example, would delete one of the bases, for example, this adenine, what would happen? We would have new sequence. So here would be new sequence. So instead of ATA, we would have ATC, and next codon would be GGA instead of CGG. So the whole sequence would be disrupted. This wouldn't be the same protein anymore. We can disrupt sequence by deletion of one nucleotide, or we can insert one nucleotide. This would have the same effect. And of course we can disrupt the gene by deletion of the large fragment. And what is the consequences? Consequences is that protein wouldn't be produced. And when we delete such a gene, or we call this knockout, as for example, if we use mice as model organism, we can then see what changes this would cause. And then we can extrapolate our data on humans. Those not necessary, this would have the same 100% consequences in human organism, but we can expect that consequences would be very similar. Of course, we cannot use humans to do such experiments, and we use model organism with short life expectancy in order to see all the consequences of such experiments. And this is also one of the ways how we find functions of the genes that functions are not known yet. So as you see, the correct answer would be answer A. This is true statement. Knockout mice can serve as model organism for study of the human genetic disorders. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.